Imagine for a moment a future where we could actually edit out the genetic root cause of conditions like Down syndrome.
It's something scientists have aimed for a long time.
Yeah, because for decades, Down syndrome, caused by that extra chromosome 21, it's been this huge challenge, mainly because we just haven't had good ways to, remove that extra bit of genetic material.
Exactly. The focus has really been on managing symptoms, which is absolutely vital, don't get me wrong, but it doesn't fix the underlying genetic cause.
Right, and that's precisely why this new research we're looking at today is so, groundbreaking.
It really feels like a leap.
It does. So today we're doing a deep dive into a study from Ryotaro Hashizumi and his colleagues. It just came out in PNA Nexus, which is, a pretty serious journal. The paper's title is a bit of a mouthful. Trisomic rescue via allele-specific multiple chromosome cleavage using CRISPR-Cas9 in trisomy 21 cells. Yeah. But what it means is they've found a new, super precise way to potentially correct that fundamental genetic issue. So our mission today is to help you understand how scientists can now target and, get this, remove an entire extra chromosome.
And the potential this opens up is pretty incredible.
Okay, so let's maybe start with the 1-minute takeaway, the really key insights from this work.
Sounds good. The absolute headline here is that researchers have successfully used a refined CRISPR-Cas9 system, think genetic scissors, to specifically kick out the extra chromosome 21 from human cells that have it.
And specific is the key word,
Okay.
right? Because previous methods weren't so targeted.
Exactly. Older methods were kind of blunt. They could hit any of the 3 copies. This new approach is allele-specific, meaning it zeroes in on only the unwanted extra chromosome. That's a massive step up in precision.
And it works well.
Yeah, the study shows it's highly efficient, surprisingly so. And crucially, it seems to be much safer for the cells themselves. They even figured out they could boost the process by temporarily dialing down the cell's own DNA repair systems.
So making it harder for the cell to just patch up the cut.
Right, and maybe the most exciting part, this isn't just about changing the chromosome count. They showed it actually reverses things, it restores more normal gene expression patterns, and improves the overall health of the cells.
Wow. Okay, so it's not just a cosmetic fix. It actually seems to, reset the cells functionally.
That's what the data strongly suggests, yeah. That points towards a whole new way of thinking about potential therapies.
Okay, fantastic summary. Let's dig into the details now. Remind us, what exactly is the challenge with trisomy 21?
So Down syndrome, scientifically it's called human trisomy 21. It just means there's a third copy of chromosome 21 or HSA 21.
And it's quite common, relatively speaking.
It is. It's the most common chromosomal abnormality that's viable, meaning compatible with life. Happens in about 1 in 700 live births. And it's the leading genetic cause of cognitive impairment.
And despite knowing this cause for a long time, the big gap has been actually doing something about that extra chromosome, right?
Precisely. We've gotten much better at diagnosis, understanding the features, supporting individuals, but methods to actually eliminate the extra chromosome from the cells, those have been, very limited. It's the difference between knowing the problem and having a tool to fix it at the source.
Which brings us to CRISPR-Cas9. Most of our listeners probably know it, but maybe a quick refresher.
Sure. CRISPR-Cas9 is basically this amazing genome editing tool, like molecular scissors, as you said. It lets scientists make really precise cuts in DNA to insert, delete, or change specific sequences.
And people have used it before to try and get rid of whole chromosomes.
Yes, by making multiple cuts on a chromosome, the idea is to destabilize it so the cell gets rid of it. But those earlier methods, they were often allele nonspecific ANS.
Meaning they couldn't tell the difference between the 3 copies of chromosome 21.
Exactly. They could cut the one from the dad or either of the 2 from the mom. And that's a problem because some genes on chromosome 21 have this thing called parent of origin effects, or imprinting.
Right. Where it matters which parent the gene came from for it to work properly.
You got it. Some genes are silenced if they came from mom, others if they came from dad, or they're just expressed differently. So if you randomly chop out one of the 3 chromosomes using an ANS method, you risk messing up that balance.
Okay, so you could potentially cause other problems, like imprinting disorders, by accident.
That's the risk, which highlights why you really need a method that can pick which specific extra chromosome to target and remove.
And that is the breakthrough here, the allele-specific, or AS, targeting.
Yes. This study introduces what they call a comprehensive allele-specific Cas9 target sequence extraction method. The whole point is to find unique markers on just one of the 3 chromosome copies, the extra one you want to remove.
But how? How do you find a unique postcode on one chromosome when you have 3 that look almost identical?
It's pretty clever. They used induced pluripotent stem cells, iPS cells, derived from an individual with trisomy 21. And they developed this technique called haplotype phasing.
Haplotype phasing.
Yeah.
Okay, what does that let them do?
It basically allowed them to figure out which specific genetic variations or alleles travel together on the same physical chromosome strand. They could essentially map out the signature of the chromosome inherited from the father and the 2 inherited from the mother, M1 and M2.
So they could distinguish P, M1, and M2, like giving each chromosome a name tag.
Sort of, yeah. They even made versions of the cells where they deleted one of the parental chromosomes, which helped confirm their mapping. It's like genetic detective work, as you said earlier.
Okay, so they have their map. How did they choose which one to target?
They made a strategic choice. They decided not to target the paternal P allele. Why? Because some genes on chromosome 21 are known to be sensitive based on maternal origin, so to be safe, they avoided P and randomly chose one of the maternal alleles, M2 in this case, as their target for removal.
Makes sense. Target identified. Now, how do you design the CRISPR scissors, the guide RNAs, to only cut M2 and leave P and M1 alone?
This was a huge bioinformatics task. They extracted all the potential sequences that Cas9 could recognize in their specific cell line's genome. Then they did this kind of comparison, a set operation, to find sequences present only on the M2 chromosome and not on P or M1.
And did they find many?
An astonishing number. They found over 15,000 potential M2 specific Cas9 target sequences. And here's something really interesting. Almost half of them, nearly 7,700, were clustered in one specific region near the end of the chromosome's long arm, the Q subtelomere.
A hotspot, even though that region is only about 10% of the whole chromosome.
Exactly. That clustering was unexpected, but potentially very useful for targeting.
So did they test these potential cutting sites? Were they all truly specific?
They selected 56 of these M2 subtelomere targets to test. Out of 54 they evaluated, 18 of them, about a third, showed perfect on-target cutting only. No detectable cuts anywhere else they looked. That level of precision is absolutely critical if you're ever thinking about therapy.
Right, you can't have scissors cutting randomly elsewhere in the genome. Okay, so they have precise tools. Now, the big test. Can they actually make the cell ditch the extra chromosome? How did they compare AS versus the older ANX methods?
They created CRISPR systems designed to cut the M2 allele at different numbers of these specific sites from just 1 cut up to 13 AS cuts. Then they made comparison systems using the old ANS approach, but with way more cuts, 24, 49, even 73 non-specific cuts. They put these into the Trisomy 21 iPS cells and watch what happened.
And the results? Did more specific cuts work better?
Dramatically better. With the AS method, the more cuts they made at specific sites, the higher the rate of chromosome elimination. It went from 1% with 1 cut up to over 13% with 13 specific cuts.
Okay, a clear dose response. What about the ANS method with many more cuts?
Much less effective. Even with 73 non-specific cuts, the correction rate only reached about 8%. And there was no clear relationship between the number of ANS cuts and the success rate. Plus, the AS method was much gentler on the cells.
Gentler. How so?
The cell survival. With 13 specific AS cuts, about 57% of the cells survived the process. With 73 non-specific ANS cuts, survival plummeted to less than 13%. It's a huge difference. The AS approach seems much less toxic.
So AS is more efficient and safer. That's compelling. And were they definitely losing the whole chromosome, not just pits?
They checked that using fish fluorescence in situ hybridization. It uses glowing probes that stick to specific parts of the chromosome. Their analysis confirmed that yes, in most cases, the AS cuts led to the loss of the entire chromosome 21, not just the end piece breaking off.
Okay, so the mechanism seems to be whole chromosome loss.
Yeah.
Now, you mentioned earlier they tried boosting this by interfering with DNA repair.
Yeah, that was another clever part. See, when CRISPR makes a cut, a double-strand break, the cell's natural instinct is to repair it.
Right, it tries to patch things up.
So the researchers thought, what if we temporarily block those repair pathways? Maybe the broken chromosome is more likely to get lost if the cell can't easily fix it. They targeted 2 key repair proteins, PLLQ and LAG4, using sirnina, which temporarily silences their genes.
Like putting the repair crew on a coffee break for a bit.
Huh, something like that. The silencing lasted about 48 hours, and it worked. Temporarily blocking PLLQ and LAG4 increased the chromosome loss rate by an average of nearly 1.8 times across the different AS cutting strategies.
Wow, almost doubled the efficiency in some cases.
Yeah, it really highlights how important those repair pathways are and maybe gives a clue about the time frame the cell uses to decide whether to repair or discard a damaged chromosome.
This is getting incredibly precise. How did they absolutely confirm it was the targeted M2 chromosome being lost and that the resulting cells were truly normal, chromosomally speaking?
They used a really neat trick. 3-color reporter cells. They basically tagged each chromosome 21 with a different color fluorescent protein, say blue for the paternal P, red for maternal 1M1, and green for the targeted maternal 2M2.
So if the cell lost the green signal,
it meant it had specifically lost the M2 chromosome, and that's what they saw. When they used the M2 targeting AS vector, about 7% of cells lost the green M2 signal, way more than lost blue or red. The non-specific ANS vector caused more random loss of all colors.
very visual confirmation.
Yeah.
Did they check individual cells too?
Yes, using genetic fingerprinting called STR analysis on clones grown from single cells. After treatment with the M2AS system, almost a third of the resulting cell lines had the correct 2 copies of chromosome 21, Trisomy. And importantly, all of those corrected cells had specifically lost the M2 allele.
All of them. That's amazing specificity.
It really is. The ANS method, in contrast, produced far fewer corrected cells, and only a tiny fraction had specifically lost M2. Then they did karyotyping, basically taking a picture of all the chromosomes. The original cells were 100% trisomic. After the M2AS treatment, over 37% of the cells looked perfectly normal, 46 chromosomes XY, no extra 21, and no other obvious structural problems. A clean correction.
That's hugely reassuring on the safety front. But what about looking even closer at the whole genome sequence? Did the AS cutting leave any unintended scars?
They looked using whole genome sequencing, WGS. First, it confirmed the corrected cells had the right dose, 2 copies of chromosome 21 material. As for off-target mutations, they found remarkably few.
How few is few?
On average, only about 5 to 7 unintended sequence variants per cell line. And interestingly, most of these weren't random hits across the genome. They seem to happen mostly at the intended target sites on chromosome 21, but on the wrong allele, like the guide RNA, sometimes bound imperfectly to the P or M1 allele at the target location. Crucially, they found basically no Cas9-induced mutations away from the intended chromosome 21 target areas.
Okay, so the collateral damage seems very localized if it happens at all. What about bigger rearrangements, structural variants?
They saw very few of those in the successfully corrected cells, maybe 1 per cell line, compared to 5 or 6 in cells that remain trisomic after treatment. And again, these were mostly located on chromosome 21. It suggests the impact is quite contained.
Given all that safety data, could they make the process even more precise? Maybe by picking only the best guide RNAs.
They did exactly that. They went back to their data, picked 6 guide RNAs that had shown 0 off-target activity in their tests, and made a new, refined M2AS/6 vector.
And how did this ultra-refined version perform?
Impressively well. It achieved a chromosome elimination rate of over 13%, which was right up there with their earlier vectors that used more guides, like the 8 guide, or even close to the 13 guide one.
Interesting. So it's not just the number of cuts, but the quality and specificity of those cuts that really matters.
It seems that way, yes. Both quantity and specificity play a role.
Okay, this is all technically fascinating, but let's get to the payoff. Did fixing the chromosome number actually fix the problems associated with trisomy 21 at the cellular level? You mentioned changes in gene expression earlier.
Yes, and this is perhaps the most compelling part of the study. They used RNA sequencing to look at which genes were active in the original trisomy cells versus the corrected rescued cells. The difference was stark.
How so?
The gene expression profiles clearly separated based on whether the cells were trisomic or corrected, euploid. They found thousands of genes whose activity levels changed significantly after correction.
What kinds of genes were changing?
They used gene ontology analysis to group these changing genes by function. What they saw was really telling. Genes involved in metabolic processes, things like making cholesterol, processing sugars, energy production in mitochondria, these were generally downregulated in the corrected cells.
Which fits with the idea that trisomy 21 cells are sometimes hypermetabolic, right?
Yeah.
Too active.
Exactly. It looks like correcting the chromosome number helped normalize that metabolic overdrive. But even more exciting were the genes that were upregulated.
Which were?
These were genes strongly linked to things like cellular development, nervous system development, making new neurons, neurogenesis.
Wow. So correcting the chromosome count actually switched back on gene programs related to brain development.
That's what the data suggests. It implies that removing the extra chromosome can restore transcriptional signatures relevant to the very neurodevelopmental differences seen in Down syndrome.
That's potentially huge. Did they compare this to other Down syndrome research?
They did. They compared their results to data from identical twins, where one twin has Down syndrome and the other doesn't. They found a really high degree of overlap, about 90% match in the top functional categories of affected genes. This gives strong confidence that what they're seeing is a genuine rescue of the Trisomy 21 cellular phenotype.
Okay, so the gene activity changed. Did the cells themselves behave differently? Were they healthier?
Yes,
Yep.
significantly. The rescued, corrected cells grew faster, their doubling time was shorter. And remember, we talked about reactive oxygen species, ROS. These damaging molecules are often elevated in trisomy 21 cells linked to mitochondrial issues.
Right.
Well, ROS levels were markedly reduced in the corrected cells. So, taken together, it shows a real functional improvement in cellular fitness and health. It seems reversible.
That's incredible. Now, a key question for any therapy. Does this only work in these lab-standard iPS cells, or can it work in cells more like those actually in the body?
Great question. They tested their best vector, the M2AS/13, in primary fibroblast skin cells taken directly from an individual. These are differentiated, mature cells.
It did?
Yes, it did. They saw a chromosome elimination rate of almost 14% in these fibroblasts, much higher than controls.
Okay, so it works in differentiated cells too. What about cells that aren't actively dividing? Many cells in our bodies are like that.
They checked that too. Even in fibroblasts that weren't dividing, they used a marker called EDU to check. They still saw a chromosome elimination rate of over 3%, still significantly above the background noise.
That's really important. It suggests the approach isn't limited to rapidly dividing stem cells and could potentially work across a wider range of tissues.
It definitely broadens the potential scope, yes. It's a crucial piece of evidence for thinking about future applications.
Okay, this all sounds incredibly promising. But no research is perfect, especially at this stage. What are the main challenges or hurdles still remaining?
Well, one key safety concern is what happens in cells where the treatment is applied. But the target chromosome doesn't get eliminated. You've still made cuts. There's a risk of leaving behind small mutations or alterations at those cut sites.
Right, even if the chromosome stays, it might be slightly scarred by the attempt.
Exactly. And while off-target activity was low, ensuring it's truly zero, or finding ways to mitigate any effects on the non-target alleles at the intended sites is still really important.
Are there other ways to approach this that might avoid cutting DNA altogether?
That's definitely an avenue for future research. Exploring epigenomic editing, changing how genes are read without altering the DNA sequence itself could potentially sidestep the risks associated with double-strand breaks.
That makes sense. What about refining the targeting itself?
Well, this study focused heavily on that subtelomere region. We need more research on whether targeting other parts of the chromosome, like the centromere, might be more or less effective or have different consequences.
And practically speaking, that initial mapping, the haplotype phasing, sounds complicated.
It is. Finding ways to simplify that, perhaps using newer long-read sequencing technologies, could make the whole process more streamlined and accessible.
And testing in more cell types.
Absolutely critical. While fibroblasts are a good start, for something like Down syndrome, seeing if this works effectively and safely in neurons, glial cells, other brain-relevant cell types is a must for translation.
What about digging even deeper into the genetic changes?
Yeah, more comprehensive WGS analysis, looking really closely across all the conditions, would give a fuller picture. Also, understanding the role of other DNA repair pathways beyond POLQ and LIG4 could be important. And fundamentally, figuring out the cellular decision process, why is a broken chromosome sometimes lost and sometimes repaired and kept. Understanding those factors could lead to ways to further boost efficiency.
And then there's the elephant in the room for any potential gene therapy.
Delivery. How do you get this complex CRISPR machinery safely and effectively into the right cells inside a living organism? That's a massive hurdle for in vivo application, but it's the ultimate goal.
Absolutely. So let's try and wrap this up. What we've really unpacked today is, well, quite remarkable, isn't it?
It really is. This allele-specific CRISPR approach offers a genuinely new strategy. It's not just managing symptoms, it's tackling the root genetic cause of Down syndrome at the chromosomal level.
And the evidence suggests that by precisely removing that extra chromosome 21, they can actually restore more normal cellular function, normalized gene expression, even in differentiated cells.
The specificity, the improved efficiency compared to older methods, the reduced toxicity, the functional rescue, it all points towards a powerful potential pathway. It shows trisomy rescue is feasible.
For anyone listening who follows genetic medicine, or just the cutting edge of what science can do, this research really feels like a significant marker. It shows how these targeted genomic tools are moving us closer to potentially correcting complex genetic conditions in a fundamental way.
It's a testament to the power of precision genome editing.
Definitely. And it leaves us with a thought, doesn't it? As we keep developing these incredibly powerful tools that let us rewrite our genetic blueprint with ever-increasing precision. What are the new ethical questions, the societal discussions we need to have about wielding that power?
That's the big question looming over all of this progress, isn't it? Something definitely worth thinking about.